Welcome back. In this recording we're going to look at another key technique, and it's going to be centrifugation. So this is a technique which is useful for separating cellular components using centrifugal force. So let's take a look at this. So the basic premise here is that you can take a isolate of cells. So you collect some cells either from a tissue or from cells grown in culture, that kind of thing, and then you can homogenize them, which means you basically smush them up with a pestle and mortar like device, and that breaks the cells open. And then you can put the contents of the cells and the cell fragments into a plastic or glass tube, and then that glass tube goes in a machine which spins at very high speeds. And because of the relative densities of different cellular structures, you can separate them based on their density. So I've put some images of some centrifuges here, ranging from small to large and expensive. So this centrifuge on the left here is a few $1,000s to put on a bench, $7,000 or $8,000 to put on a bench. It's called a benchtop centrifuge, and you can see it takes little small tubes in this centrifuge head. These are small 1.5 milliliter tubes, little plastic ones with those pop caps that you might have seen in the labs, and then a lid goes on here and the lid gets closed, and it'll spin samples up to about 20,000 G. Now right now you're experiencing 1 G. That's the force exerted on a mass by the Earth's gravitational pull. So that's 1 G. That machine on the left can spin things up to 20,000 G, so that's a huge amount of G-force that's being generated. The next one here is a larger desktop or benchtop centrifuge, and this will spin bottles and flasks. So if you've got E. coli growing in a bottle or something, you put them in a bottle, you can spin the larger bottles here. Now this one won't go as fast, but this one's often refrigerated. These smaller ones can be refrigerated. This one looks like it's refrigerated because it's slightly larger, and these ones can be refrigerated or not refrigerated. So these are going to spin at lower speeds, maybe 4,000 to 5,000 G, so not as fast. And this beast in the middle here, this is called an ultra centrifuge. So this will spin tubes and exert the contents up to about 200, 300, 400,000 G. That's 400,000 times the force of gravity. So the volumes that are being spun here are very small. We're talking about anything from a milliliter up, so that would be a gram, all the way up to maybe 15, 20 grams in some cases. So you can imagine the amount of kinetic energy that's being generated when this thing spins at 300,000 or 400,000 revolutions per minute. Your car engine goes up to about 8,000 if you redline it, and so think how fast this is going at 200, 300, 400,000 revolutions per minute. So when these machines aren't operated properly, things like this on the right happen. So what can happen is if you don't have the two sides of the centrifuge head balanced exactly, we're talking down to 0.01 grams here. If you don't have the two opposite sides of the centrifuge head, which is where you put your samples, balanced, then the centrifuge head doesn't sit square on the spindle on which it spins, and so eventually, as it spins up to speed, the head can come off and just catastrophically destroy the machine and the lab equipment around it. So these are extremely expensive, and you have to have some technical competence to operate them. So down at the bottom here, we're looking at sorry, this isn't differential centrifugation. This is just the general principle of centrifugation. So ignore differential centrifugation. I guess it is a type of differential centrifugation, so that's okay. I guess we can live with that. So imagine this tube here is a extract made from some cells. So we've taken some cells and we've grown them in dishes in the lab, and we've homogenized them up, and we're interested in isolating just the cytoplasm. So we've smushed them all up, broken them apart, all the mitochondria, all the peroxisomes. Those have all been released, and so they're all floating around, and each of those cellular components, we've got nuclei in there, they have different relative densities. So the purple balls are going to have the biggest density, and then the blue balls will have lower density, and then we've got small particles here, little black dots with even lower density. So imagine we spun this at, let's say, 500 G. the things with the large relative density would start to sink. And now if we spun it longer or we turn the speed up, let's go up to 2000 G, now the large things have pelleted, so that's called the pellet at the bottom of the tube, and now the intermediate density items or cellular components are starting to pellet. If we increase the speed, and now we're starting to pellet the smaller things, increase the speed, the large high density, intermediate density things are completely pelleted, and we've nearly pelleted the smallest or lowest density material. And then if we turn the speed up again, we can pellet everything, so we're pelleting all the cell fragments, all the membrane fragments, nuclei, mitochondria, lysosomes, Golgi fragments, all that stuff, and then we could be left with just the cytoplasm here. And so if we wanted to study some kind of chemistry going in the cytoplasm, we can isolate just the cytoplasm, and then in the pellet would be all the other components. So the faster you spin it, the more material you pellet, because of the range of densities of material you can centrifuge, you can pellet at higher speeds. So another variation of differential centrifugation is what we're looking at here. So same kind of thing at the top here is what we're doing is we're mushing our cells up, and usually do this on ice on a beaker. So we smash the cells up, so we've kind of homogenized them and broken them up. And then if we take that cell extract and we spin it at 1000 G, that's going to capture in the pellet the nuclei and any cells which haven't been broken up by our homogenization. And then everything that's not the nucleus or an intact cell would be in the cytoplasm. So in there we've got our mitochondria, our Golgi fragments, all that kind of stuff. Now if we take that supernatant, which is the non-pelleted material, and we spin it again up to 20,000 G, this time we could pellet out things like mitochondria, lysosomes, peroxisomes, and whatever's left would be in the supernatant. So now we could isolate the peroxisomes, for example, and the mitochondria. If we wanted to study a mitochondrially enriched extract from cells, we can harvest that. If we take the supernatant from that and we go up to 80,000 G, now we're looking at pieces of membrane, ER, that kind of stuff. And so we could take those fragments and we could biochemically and structurally analyze those if we needed to. If we take that supernatant and spin it again up at 200,000 G for a couple of hours, now we're getting things like ribosomes, viruses, pieces of DNA, that kind of stuff. That would all start to sediment now. And so this is differential because as we increase the speed and we isolate the supernatant or the pellet, we've isolated an enriched fraction. So if we want to study the mitochondria, we know that we can spin the cellular extract at a certain speed, take the pellet, and that's now enriched in mitochondria. So this can often be the first step in isolating a specific protein. So if we're interested in studying cytochrome oxidase or NADH dehydrogenase or mitochondrial proteins, then what we can do is enrich a cell extract from mitochondria, and then that's our first step in purifying a particular protein, because we've removed everything else via this process of differential centrifugation. Now, a neat and kind of cool trick you can do with this is something called density gradient centrifugation, and this is really good for isolating all of these different cellular fractions in one kind of go, so you don't have to do these stepped up speeds and that kind of thing. So let's say we want to isolate mitochondria and lysosomes to study some reactions they're involved in. So what we're going to do here is we are going to, in a tube that's going to go in our centrifuge, we will create a gradient of something. So this could be something like glycerol, or it could be sucrose. So the way that you do this is, let's do it with sucrose. You take a solution of, say, 20%, maybe 40% sucrose. So you dissolve 4 grams of sucrose in 10 milliliters of water. That gives you 40% sucrose. And then you put it in your centrifuge, and you spin the centrifuge at very, very high speeds for a good number of hours. Well over 2 hours. And what happens then is that you start to actually even sediment the sucrose molecule. So what you end up with is, at the top, lower density solution, and at the bottom, higher density solution. Then what you do is you overlay on top of that gradient a cell extract or a mixture of whatever it is you want to try and separate. So imagine this is an extract which we've taken out of a tube which has been subject to differential centrifugation, and we know that it contains mitochondria, peroxisomes, and lysosomes, and we want to get hold of, let's say, the mitochondria. So we're trying to get rid of all the other stuff. Now, those three organelles all have different densities because of the relative different sizes, different volumes, that kind of stuff. So as we layer this on top of this sucrose gradient, remember, dense at the bottom, less dense at the top, and then we subject it to, again, very high speeds of centrifugation, is what will happen is that the denser or heavier particles, if you like, will start to sink faster during the centrifugation than the lighter things. The lighter things will stay on the top. Because these things all have different relative densities, as we spin the tube over the course of several hours at very high speeds, the lysosomes will separate out from the mitochondria, will separate out from the peroxisomes. And what we can do, starting at the top, we would stick a needle in the side of the tube here, and we would suck out a small volume. And so we would keep taking out small volumes from the tube and putting them in new, smaller tubes. And then we could do some biochemistry to look to see which fraction from our tube here contains the mitochondria, and so we could run the proteins, we could run the extract, the sample from this tube on an STS polyacrylamide gel, like one of the ones we talked about very early on, to look for proteins which we know are localized in the mitochondria, and that helps us isolate then specifically mitochondria if we want to do some biochemistry or we want to do some structural studies on the mitochondria. So that's a really neat little twist on centrifugation. We'll wrap it up there. And when we come back, we'll talk a little bit about some organelles which you may not have heard too much about in the past. So I'll speak to you soon.